So welcome back, everybody. And in this session, we will have a kind of artistic aspect about the biomedical visualization. So our first speaker is Drew Berry, and Drew is a biomedical animator at Walter and Eliza Hall Institute of Medical Research in Melbourne, Australia. If you have ever learned anything about molecular structures through animation, you probably already watched a lot of his videos. So Drew creates many beautiful, accurate visualization of dramatic cellular and molecular action that is going to inside our body. I would say this is the most enjoyable way to study biology. As a cell biologist and an artist, he works as a translator, transforming abstract and complicated scientific concepts into vivid and meaningful visual journeys. His animation has exhibited at a lot of museums, MoMA, V&A, and Royal Institute of Great British and the University of Geneva. And Drew will lead us to see this beautiful world of BioMedVis approaches. So let us welcome you, Drew, and the floor is yours.
Thank you very much. It's a great pleasure to be presenting, although from the other side of the world. So hopefully we won't have too many technical difficulties. So hopefully you're seeing a building with a large column of colored light. That is where I work. It is Australia's sort of most prominent and best funded medical research institute. And they do lots of research into various diseases, cancers, infectious diseases like malaria and autoimmune diseases. And I've been there resident.
Sorry, Drew, maybe you want to share. Maybe you want to share your screen again because we are losing your slides.
Oh, you're not. That's interesting because it's working from here. Okay, I'll go back into Zoom. Thanks for letting me know. All right. So, what do I see? Yep. Share. Try this again. So I've been resident there as an artist, science artist, for about more than 25 years. And what I do is, my background is in cell and molecular biology, in microscopy. And so I'm able to read and navigate the literature, and I access all the various forms of data, do a large literature review, start a very project, and reconstruct as best I can, as accurately as I can, the science that's being represented. So I'll just finish on this column here is one of the large artwork installations I've done. And this is actually, it's displaying a nonstop roster of raw images from the imaging lab and structural biology and so on. And the idea is to get people to think about what's going on inside the institute. What I'm going to talk about today, hopefully I can get rid of the bar at the top, doesn't matter, is I've just completed a 3-year project doing on respiration. So the process of how we extract energy from glucose from the food we eat, and also oxygen from the air we breathe. And I have covered the entire enzyme pathway, all the way from glucose to generation of ATP, which goes on topics like the citric acid cycle, electron transport chain, and glycolysis. So these are all diabolical topics that are usually taught. These are just some images from the whole set of animations. It's about 30 or 40 minutes total of animations. I'll play a couple of them, but I just wanted to show you some images of them because I expect that these would be eaten alive by the compression over video. But the link at the bottom there, the YouTube link, is to an unlisted collection of the animations, so you can view them before they're published. They'll be published in the coming weeks. So the animation I'm going to talk about today is the 5th enzyme in the electron transport chain. So this is a figure here from, you would typically see in a textbook or in a review paper in literature, representing the various enzymes in the electron transport chain, a part of the whole respiration process. And it starts on the left, and through a various chemical enzyme reactions, we end up to the enzyme on the right, the ATP synthase. And usually students have to learn all the chemical names, and it's a rote learning and very, very hard to understand process. And I've gone through every enzyme and reconstructed from the PDB, from cryo-EM, and from dynamic simulations, the entire pathway. We're going to talk about complex 5, ATP synthase, on the right-hand side there. And what it's all about is the generation of ATP, the chemical fuel that powers all life on Earth. This particular animation is actually a simulation, molecular dynamic simulation created by Mike Kuiper, who's here in Melbourne. And I've collaborated with him on many things. So this is a small molecule simulation to be surrounded by water molecules, which we've removed so that you can just see the ATP itself. I can incorporate this and many other forms of data into the animations to make them come alive and be as accurate as I can. So when you see the small molecules like this in the animation, they are all from simulation. This is how ATP synthase is typically represented in a published paper. You have the figure on the left is an illustration. In the middle, you have from cryo-EM as well as protein data X-ray crystallography. And then on the right is an animation from the paper, from a figure in the paper showing the dynamics of ATP synthase. So ATP synthase is a very popular topic and it's been done many, many times before. But I thought I could bring some new perspective to show the dynamics and the nature and the landscape of where ATP synthase actually occurs is kind of my goals for this one, showing the dynamics of it. So this is a early model. This is from early 2018. It's a dynamic model from a combination of protein data bank, X-ray crystallography, as well as an emulation. It's not a simulation of the chemistry that's going on there. So this just as a simple model. This is a representation of ATP synthase actually working at real time. I like to incorporate that into my animations where possible. So producing 80 to 100 ATP per second. So it's something it's certainly watchable. It's not unimaginably fast. Just to briefly, some of the sorts of data that I always do a review of literature, and this is from a paper where you're starting to see ATP synthase forming these pairs embedded in the membrane of inside mitochondria, and they cause this fold in the membrane. And also on the right-hand side, you see where they've placed the two X-ray crystallography models, and in the middle is all the measurements and such, and I can incorporate that directly into reconstructing animation. This is from a figure for use in classroom use, reconstructing the dynamics and trying to show some of the chemistry. And the main issue is you have the membrane being folded there with a very high concentration of protons, hydrogen ions down the bottom, and versus the outside. And this is kind of the motive force that's going to drive the rotary engine of ATP synthase. This figure was done, it's provided for students as an interactive where they could touch and it labels different parts and so on. The animation I'm about to play, it was quite a fun one, reconstructing ATP synthase. Part of it you'll also hopefully hear is the musical score. It has a choir song, and part of the vision and attempt for this particular animation is I wanted to have a female narration versus what I usually do is my own narration, and also have a young voice. And so that it could relate to high school biology students as well as university level. And the sound score, so this is from the school where my students, my kids go to, Carey High School here in Melbourne, and we had the middle school choir singing part of the background music for this particular animation. And the gentleman on the right there with the blue t-shirt, he's Frank Titaz. He does all my sound design. He has since school days, and he's a multi-Grammy winning artist doing sound design for film and music. So here, this is hopefully going to work. I'll play the animation. It just runs for 2.5 minutes about ATP synthesis.
All life on Earth depends on this tiny, energetic molecule, adenosine triphosphate, or ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA. Generating ATP for your living cells are mitochondria, electrochemical batteries that converts energy from the food you eat and oxygen from the air you breathe into ATP. So where does your ATP come from? Deep inside your mitochondria, rows of molecular motors generate ATP. the molecule essential to all life on Earth. Enzymes bring together reactants to form a chemical bond, converting mechanical energy into chemical energy. A ring of enzymes work in step, creating 3 molecules of ATP with each cycle. Inside the molecular motor, a rotating axle powers the sequence. The axle is attached to a rotary molecular motor, moved by the force of protons pushing from the other side of the membrane. A difference in proton concentration propels the molecular mechanism. Synthesis of ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA.
So I'm not sure how that would come across over compression over the internet, but so again, you'll be able to download these. All of these animations are available for free under Creative Commons license, and they will be translated in different languages and provided in various ways. So this particular animation was designed as a hook to introduce people to the topic and to bring them in and to engage them. There are many more serious and more classroom-based versions. That animation I just showed you was actually created in 2018, and it was the technology we were using was originally inspired and by Ivan Viola and his cell view system. We've now created our own pipeline, but it's very much the original inspiration was Ivan Viola's system. So because it's all in game engine, we're now able to easily generate, as you're seeing here, like augmented reality type visualizations, virtual reality visualizations, and real-time explorations of these things. But all of that identical model I was playing in the animation, which is a combination of simulation and emulation animation, works dynamically, easily on an iPad, and which is staggering. So we're really very interested in mobile devices and getting access to students that way. We're also collaborating with in Norrköping with their 8K beautiful domes, which I wish I was over being able to visit right now, but that's not to be. But we're now creating all of these animations, or have created creating these animations for 360 dome experiences. This is actually in a dome here in Melbourne, a mere 6K dome that we have here in Melbourne, and just an early test. All of this work was from about 2018. So in last year, 2020, of course, the COVID story, of course, came up. It's one when I personally wasn't keen to get too quickly involved with, just because I knew there's so many great people around the world who would be doing great COVID graphics. But then one of the researchers at the Institute did some significant work in developing custom antibodies. And so I had to quickly generate some graphics for that. And so I just want to briefly talk about what went into that. One of the things I wanted to represent, which at the time, this is from August last year, is a lot of the COVID visualizations or representations were vivid of spheres, rigid spheres, and where if you look at the electron microscopy, you can see that they're very dynamic and can change shape in various ways. So I wanted to capture a little bit of that. This is again from Mike Kuiper, who did my other simulations. This is just the heads of the spike of the COVID on the COVID virus, and this is actually a glycosylated or a covered in sugars spike. This is a molecular dynamics simulation, and we're able to take that data and with our game engine technology make lots and lots of representations. So the COVID virus is covered with this dynamic spike. So this is sort of data that I look at to reconstruct the structure, but also seeing how the socks are very flexible, and where they're flexible, I can reconstruct that in animation. These are the antibodies and nanobodies that the researcher I was working with was using, but they of course were provided as static X-ray crystallography models, and I articulated them and made them dynamic in animation, and that's going to be incorporated in the animation. So yes, so the software I'm using is Maya, is my animation software, where Maya is very good, and we have multiple pipelines now for bringing in various forms of data. We then bring it and combine all the various forms of data in Maya, and then we export to Unity for our real-time visualization. And we're currently rendering 8K 8K rendering, 8K frames, 8K resolution is about 1.5 seconds per frame. It's just a profound change in our productivity at the moment. And we are also exploring and doing some VR and other work like that. This is one of the visualizations from August last year. This is so of the dynamic virus. I've got the membrane outer coat, and then you have the stalks, having their articulation and dynamic behavior, and then you've got the antibodies fluttering in like little butterflies and attaching. And so the considerations of this are very simple ones, like color. I'm using greens and yellows to represent the disease, the virus, pinks and reds representing the heroes here, which are the antibodies. So when you get a vaccination, your body will generate antibodies, and they'll attach to the virus and block its entry into the cells, and also flag it for removal. This is one. It's a little bit exuberant, but this is they're interested in creating cocktails of antibodies, and so that's what this representation is for. This is really meant for just media release, for promotion, and for public outreach. It's for popularizing the topic rather than being accurate, too accurate in a representation. So that's close to it. This is my small team. Justin Muir in the middle there is from 20 years in the games industry. He's completely transformed our workflow and pipeline over the last 3 years. And if you look at our my recent animations, which I think I would link next, you'll see the power of what he's brought to our capabilities now. Etsuko has been with me for 13 or 14 years. She's a similar background to me. She's a scientist who's transformed into animation, and she works on topics within the Institute. She's doing a ubiquitin animation, and she recently finished one on P56 last year. And okay, so if you wanted to see my respiration animations, and this is a sneak preview, this is not publicly available, or some of them aren't, like glycolysis and pyruvate. If you go to tinyurl.com respiration 2021, that'll hopefully take you to our YouTube, our hidden YouTube playlist. That's the best way I can deliver them right now for you, but they will be available for free downloads, hopefully in the next week or two. And so with that, I'm happy to take any questions. I think I probably went over time.
Questions from the chat. Let me begin with, so Heavy Couser, Heavy is asking, thank you for the great videos. So his question is, I found and find it highly interesting to think about how to convey process that happen at largely different time scale. One example that we found that we focus on, for example, is polymerization, where nanoseconds meet seconds across the entire process. Besides accelerating the animated display, what other approaches do you find useful to cope with that temporal multi-scale challenge?
It is always a challenge because a lot of these reactions, even within the same system, happen at such magnitudes of different speed. And I'm under the constraint and driven by, this has got to be a meaningful communication, and you only have a certain amount of time for people's attention, and you need to tell the stories in a succinct and meaningful manner. So I tend to acknowledge and recognize the different speeds of things, but I need to bring into a framework so it's visible and watchable for humans. It plays at 30 or 60 frames a second. So if it's a nano, or femto scale, and it's just not imaginable, let alone. So that's one of the compromises I certainly do, is combine things that are at different rates of speed, but that's not the priority generally of what, or the main meaning purpose of the animations, which is more to show you structure and the dynamics in general, and to convey the location, the setting of where these sorts of reactions are happening, to make it memorable in that way. So it is a challenge speed, but wherever possible, if the speed is meaningful to humans, if it happens at a rate that we could watch and see, I will certainly incorporate that. And I do in many of my animations. It's a moment that is very universally popular, is where you say, this is the real-time speed, this is how fast it operates. Students and the public really like that kind of information. So where it's possible.
Great. I see that there are questions is like exploding like crazy. So I will just follow one by one. Our next question is from Alexander Vish. The question is, how do you choose the colors for the molecules? Is there a system to it, or do you rather go with whatever seems to look good?
No, I've long time followed sort of a what works for me is, and it's to evoke a feeling, to get you to convey meaning, is why I use color. So if it's a disease thing or a parasite, I'll use yellows and greens or colors to suggest disease and things not being well. If it's a healthy living thing or our hero immune cell, I'll tend to use pinks and reds, just because as mammals we like those sorts of colors as warm colors. And then and blue colors and cool colors are always for death and dead things. But at the same time, I'm also last few years we've been switched to being very conscious of colorblind students. 7% of males are colorblind, and so a lot of my color palette is now skewed towards, so having yellows tending to be the hero object, because everyone can see the yellows, against blues for the enzymes, just to make sure that the highlight colors are there and working for everybody. It's a real challenge to work that way, but I'm sorry, like the ATP synthase I just played there was the first time I actually was switching to colorblind palette, and I don't think I succeeded in creating something that was pleasant to look at, like for me, but it worked for colorblind people. Subsequently, like if you look at the citric acid cycle and the electron transport chain, glycolysis particularly too, I'm getting better and better at managing colorblind palettes that work, I think, for everybody.
Great, thank you. So we have technical questions from Goklita. Is there any way of bringing trajectory data in Maya, or just importing multiple PDB files?
So a lot of our work is simulation, from molecular dynamics simulation. We have a pipeline specifically for bringing in that kind of data into Maya, but we don't. We bring into Maya, and we're able to create a proxy object that contains all of that data. So I'm not actually seeing that data in Maya, but then in Unity, in the GPU, it's able to play those dynamic simulations and place, like the COVID spike, every spike contains that simulation, the 300 frames of simulation data. And we're doing a lot of that kind of thing right now. So, and other forms of data. So with my programmer, Justin, being there, he has created multiple pipelines for the various forms of data that I want to bring into Maya and prepare for export to Unity.
Thank you. So we have next question from Sam Schwartz. Can you speak more about how you distinguish between emulation and simulation, where on the spectrum
Sure.
of human artists construct animation on one end and pure computer simulation on the other end? Do you find that most of your dynamic visualization today lie?
So all of my work, wherever possible, I will use real data, and if it's accessible and if it's watchable, often it can't be. It's very sometimes noisy and messy. So I call simulation is molecular dynamic simulation, the compute of articulations of how molecules internally are, their electrostatic forces, all of that. So and wherever possible, I want that, and or other forms of data, microscopy or cryo-EM. If it's available, I will use it. But then there's emulation, what I would call emulation, which is my animation techniques, where I'm creating the look and feel of what you would find in a molecular simulation, but it's under my control, and I can tell a story and I can reveal things that I want to, or emphasize different parts of the visualization. So yeah, it's I call it emulation, because I'm emulating the look and feel of what a simulation will look like, but it's vastly quicker to create and control and tweak. And if you look at citric acid cycle, for example, or electron transport chain, the small molecules are all from simulation, MD simulation. All the rest of it is a emulation I've constructed with various dynamics engines within Maya.
Thank you very much. Our next question from Ruta Shin. You mentioned about the visualization in VR. Do you think the system with those animations can provide enough rendering performance for VR? Is there any limitation regarding the rendering for VR?
Well, I've been actually quite against VR for a long time, just because of all the cables and you look goofy, and it just wasn't. It was an okay experience. I could see where it was going, but I just didn't think it was a totally captivating experience. However, now with with new mobile phones that are out now, and I'm really enjoying the Oculus Quest 2. It's just a staggering piece of technology, and we have not had any issues. It's with ATP synthase, which has 300,000 atoms on a phone, kids' phones. You can actually do AR, but you can not just create one, you can create 10 in a row, and each one is spinning and rotating in its own, has its own, like, it's just there's huge untapped power now in all this technology, and I think there used to be a limit, but I'm wondering about. I've not, the limit I thought was there is not there anymore, and we're gonna have to find out what that is. So no, we're having a great time with VR. It's working out really well.
Cool. So we have really a lot of questions. So our next question is from Stefan Buchner, and he was saying, thanks for the great talk. His question is, in terms of tools, what do you think is the area, what would be in most need of improvement from your point of view? What type of innovation could significantly improve your pipeline?
So we've just gone through a very long 3-year process of that. And right now, I can't tell you how staggered I am by the capacity and capability of our current pipeline. And it's really caused me to have to rethink what I want to set out to achieve in these visualizations, because it's well beyond what was at all possible before. But it's also responding to the huge tsunami of data and discoveries that are going on with all the different microscopy and other X-ray and cryo-EM is just fantastic. So there's so much data now to try and represent. But what we're really striving for with our system is to be able to create virtual landscapes of the interior of a mitochondria, for example, with all the various enzymes there, all live. And so again, if you go look at the pyruvate dehydrogenase animation, that will show you like the interior of a mitochondria. This is all playing live on a normal game PC. It's like a normal PC a teenager would have in their bedroom, and it's playing these sorts of worlds, these intensely complicated worlds, molecular visualizations, and it's live, it's real time, and I'm just staggered by that. I sort of catching my breath, because we've been working towards this for a long time, but it really works. It's amazing.
Thank you. Okay, maybe we
and
can
again,
go.
actually, it was inspired and very much working with Ivan Viola and his team and their CellView and being able to do this real-time GPU rendering. But we've now taken it further for our own system, as well as developing more pipelines for more kinds of data into it. And but it's just early days, but I'm really staggered by it. Sorry. Yeah.
No problem. So we have 1 minute, maybe a quick question from Sutter. So he was mentioning that they found interesting amongst I mean, they found interesting for 3D models in VR, but they view VR as novelty or useful for feeling public engagement rather than being truly useful to research. How do you think VR will fit into future scientific research?
Yeah, I'm not sure. I think it's a wild frontier of possibility. I think I think it's I haven't seen the scientists I work with haven't really got engaged with VR and the experience. It's nifty. It makes great photographs for media, but I'm not sure if it's, but as it'll get easier and easier to do this kind of thing, and as the technology becomes, it's everywhere, like for example with mobile phones doing augmented reality, I think it's a new landscape. It's a new way of us interacting with this kind of data. And for me, it's really tremendously exciting, because before I have been creating animations, which are a certain duration, let's say 2 minutes long, and you watch the animation, it's done. Where now you can light it up and the world is live and it'll play endlessly. And you can take all the time you want. It's not a 2-minute animation, it's as long as you need. And you can explore and you can go over and look at all these different objects and interrogate them and have it all light up with labels, and that's all possible. So it's a really different way of navigating, experiencing this kind of material. And so I have been very against VR, but now I'm totally sold now. I think it's a really fantastic, very makes a great impression to experience it that way in 3D.
Thank you very much.